Imagine taking a single stem cell, placing it in a lab dish, and watching it multiply until it triggers a positive commercial pregnancy test. This scenario highlights the central ambition of a new field of biology. To understand why, we have to look at the current reality of human reproduction. In vitro fertilization, or IVF, has a success rate that plateaus at about 30%. The vast majority of embryos, whether conceived naturally or through IVF, fail to implant or collapse a few days later without any identifiable medical cause. This monitor shows how fertility doctors can carefully observe and select early-stage embryos while they are in the lab. But once that embryo is transferred into the patient, all visibility vanishes. We have a massive medical blind spot during the exact weeks when human life is at its most fragile. Biologists call this post-implantation period the black box. It is the critical window where a hollow sphere of cells organizes its tissues and begins the early formation of internal organs. Studying this process hits a hard legal wall. Bioethics law prohibits growing human embryos beyond 14 days. Because we cannot observe development inside the uterus or past this 2-week lab limit, scientists cannot see why early pregnancies fail. To look inside that black box, researchers realized they needed a workaround, a model that behaved like a human embryo without meeting the legal definition of one. Solving the mystery of early pregnancy loss required the creation of an entirely new biological entity. By the summer of 2023, the work of 2 international research groups culminated in a shared breakthrough for synthetic embryology. One team was led by developmental biologist Magdalena Zernicka-Goetz, operating out of the University of Cambridge and the California Institute of Technology. The other team was led by biologist Hanna at Israel's Weizmann Institute of Science. Both teams utilized pluripotent stem cells, the blank slate cells capable of developing into almost any specialized tissue type in the human body. These institutions shifted from observing natural development to constructing it from scratch, using only stem cells as their building blocks. The process relies on self-organization. Scientists place these naive stem cells into a specialized fluid medium, where the cells use their own internal genetic code to drift together, differentiate, and physically interlock into complex structures. Nature provides the assembly manual. The scientists simply provide the environment for that code to play out. Hanna's team achieved a major milestone when their synthetic cell aggregates grew to a developmental stage equivalent to a 14-day-old natural human embryo. This cross-section compares a natural day 14 embryo with the synthetic model, highlighting the extra embryonic cells forming the yolk sac and placenta. This synthetic structure even produced the specific chemical markers detected by commercial pregnancy tests. Testing the surrounding fluid yielded a positive result. To remain within ethical boundaries, researchers engineer the starting stem cells to disable specific genes. By removing the genes required for brain and heart development, they ensure the models remain permanently incomplete. This engineering created a synthetic biological architecture sophisticated enough to trigger a biological pregnancy test. These structures are not babies, and they cannot be grown into human beings. Replicating a full 9-month pregnancy outside the physical uterus is biologically impossible with current technology. The maternal environment is too complex to simulate. Biologists describe these models as a trompe l'oeil, a convincing illusion that mimics an embryo just long enough to study its failure points. Their value lies in serving as a disposable sandbox. They allow scientists to experiment on the precise mechanics of implantation without destroying viable natural life. Researchers use these models to investigate the specific biological failures that prevent successful pregnancies. This breakthrough creates a regulatory challenge. The 14-day rule was written for embryos created via fertilization. Because these synthetic models are built entirely from stem cells, they exist in a legal gray area. An ethical paradox remains at the core of this research. The stem cells used to build these synthetic structures are often derived from real human embryos, donated and destroyed during standard IVF procedures. Organizations like the International Society for Stem Cell Research are now scrambling to rewrite global bioethics guidelines to encompass these human embryoids. In the effort to solve human suffering, we have engineered a biological entity that is moving faster than our legal definition of life.